And I think I may have said to you that during this maturation, that's when they're going to start expressing either their B cell receptor or T cell receptor, and we'll talk about that in more detail in just a moment, what that is. And then part of that training is to not attack self. And so, or not respond to self, because a B cell doesn't exactly attack. The thymus is even better at this than the bone marrow. So the B cells you'll see are mostly going to be confined to the lymph nodes and to the spleen, but T cells can actually be found in the blood as well. So they will be in contact with just about anything in the body. And so the thymus expresses every protein throughout the body. So I think I said this last time. Did I? Okay, so that's just huge. So the thymus doing this. So it's very important in the development of that T cell. Upon stimulation, that's when the B cell and the T cell get going and do their particular functions of either attacking a foreign pathogen. That's what the antibodies that B cells will produce, that's what they do. But the T cells will become effector T cells, which have other functions. And though there are in fact 3 types of T cells, T helper cells, we talked about those already when we were talking about HIV. There's another arm, the cytotoxic T cells. These will be the ones that actually kill infected body cells. And there's regulatory T cells, and they're very important in kind of like modulating the response, because our immune response can be quite large. And some of you during your oral reports will tell the rest of us that it's not so much the pathogen, but the body's response to that pathogen, which is the problem. And so regulatory T cells are in fact very important in that process, but as that's more about the immune system rather than what is happening to the pathogen, it's too much for micro. Okay, that is a huge topic in immunology, but it's beyond our scope. We just need to know about the T helper cell and the T cytotoxic cells, okay? All right, so the adaptive and acquired immune system is really about getting those T and B cells ready, and they're just waiting for antigen to be presented to them. And I've defined antigen for you already as an antibody generator. Once the B cells are challenged, they will undergo some proliferation. It's going to be called clonal selection, and they'll end up making antibodies. But the T cells, they will get stimulated, and we will turn on the cytotoxic T cells to mediate cell immunity, which means to kill an infected body cell. So here isn't just a cartoon image of 2 different types of pathogens. So we have streptobacilli and we have some cocci. These are 2 different organisms. These would elicit responses from 2 different types of B cells. These are B cells, but each one will have a specific kind of B cell receptor on its surface. This one has blue, this one has red. You see that the blue ones will make and secrete antibody, these blue antibodies, and they're after the bacilli. But the red one is secreting red antibodies against the cocci. So it's specific. Remember that we said that one of the characteristics of this leg of the immune system is specificity. And the specificity is because of shape. And so these antibodies will have particular shapes, and they'll only respond to particular kinds of pathogens. Okay? It's not a blanket response. And we'll go on to describe what it is that antibodies actually do, and it winds up causing the destruction of those pathogens. Okay. But in cellular immunity like this, here you see a cell that's infected with, looks like a naked virus. Okay? And so that cell, because it has a nucleus, will present on its major histocompatibility complex type 1, MHC type 1, a little piece of what it's currently making. And if it's currently making things like capsid or replicase, that's not right. That's not cellular. They will present that Okay, and well, this one is, they're trying to show you an antigen presenting cell. So let me take it back and say that this is a macrophage that has swallowed this and will present on its MHC type 2 to the T helper cell. Look at what I just ate. And the T helper cell speaks with cytokines, of course. And those cytokines will lead to the proliferation, which just means division, of T cells, and some of them will mature into T cytotoxic cells that will kill infected body cells. So the T cell response is actually very potent because it's directed at self. It should be infected self, but it is directed at self. And we know there's such a thing as autoimmune disorders, and unfortunately this is what's happening, is we're having an inexplicable destruction of self. That shouldn't happen, but it does. Okay, so inside the bone marrow, where we start with the initial cells that begin to divide into either red blood cells or white blood cells, and even among the white blood cells, they're kind of just waiting for a specific kind of cytokine to tell them which way to go. When they do become lymphocytes, there's 10 to the 14th to 10 to the 18th different potential B cell receptors and T cell receptors that could be made. That's a lot. This is just theoretical, but that's 100 trillion, okay, to, I don't know what 4 times that is. I don't know these numbers, they're too high. So this that is expressed here on its surface, an Ig, this is an immunoglobulin, this is an immunoglobulin D, as in dog. It's also known as the B cell receptor. Okay, so that cell is now expressing a particular IgD, and the B cell right next door to it is also expressing an IgD, but it looks different. Okay, they're not going to be alike. The same will happen for the T cell, that we will have multiple different types of T cell receptor on the surface. Once they mature and they pass, they're not responding to self, then they go and just live in the lymph or the spleen, and they're just sitting there waiting. Okay, so the B cell receptor or the T cell receptor has variable regions up here. Those are the areas that change. We've already talked about this in ELISA, how the FC region, the stem, it's going to be conserved within a species. It's the variable region that changes. And we have to remember that that just means which amino acids are there. It's going to be different ones. And they're not all the same. Some are polar, some are non-polar, some have big rings, some have little rings, some have a self-hydral group. Like, there's different ones. And so they're going to look a little bit different up at the top. And the same will be true for the B cell, the T cell receptor. On the surface of the B cell, they will have 100,000 copies of that same IgD. So in a moment, when we talk about how we stimulate a particular B cell, is that an antigen will land on its IgD. This thing is covered in IgD. If there is an antigen that's going to swing by, it will be caught. There's a ton on there. There's not quite as many T cell receptors on a T cell, but it's still in the thousands. That's a lot. This isn't at all depicted correctly because, you know, you can't really show the detail without making it a little bigger, and then that's giving us just a weird kind of understanding of these numbers. Remember, proteins are so little that certainly you could have that on the surface of a eukaryotic cell. Sure, no problem. Okay. So there's tons of different types of these receptors, and this is because that we are diploid. You have copies of how to make this B cell receptor and T cell receptor from your mom, from your dad, and they mix and match. That's what's going on here. So you might have heard a long time ago, if you took Bio 10 here, I'm not sure if they're still showing this movie about evolution, okay? But something about how they had done this study on young women and young men, and they had the young men sleep in t-shirts. same t-shirt every night, okay? And in the morning, they would put it in a Ziploc bag. Next morning, put on that same t-shirt from the Ziploc bag so that it would get infused 1 whole week. Then they took the Ziploc bags and they put them out on a table and had the young women come in, which one smells best? Invariably, they chose the smell from someone whose immune genes were more diverse, further away from her own. This is a weird thing, like we're not used to this. I mean, we feel like we can barely smell, you know, I mean, they say that all the time. Your dog is like great at this and you're like kind of, right? I mean, what happened? It's still there, it's still in us. In order to have good offspring that are going to be able to survive, you want lots and lots of variety in how many different kinds of antibody can you make? How many different kinds of T-cell receptor can you make? It also helps explain why your own siblings' sweat socks smell awful to you. Too close, okay? I mean, to someone else, right, it smells fine, not to you. That's all there is to this conversation. And so we've had this conversation a little bit about antibody structure, I think, or did I say I was skipping it till today? I can't quite remember, because in the ELISA, I felt like it was too soon to have this conversation.
I think you skipped it. I think
Okay,
you talked a little bit and then you're like, and we'll go over.
Yeah, all right, so if we can look at it a little bit more carefully. This is never depicted like this in any cartoon image because it gets too busy. But you can see that it's not just a Y, there's also, you know, first off, a Y only has 1 line for the stem, right? You see how there's 2, okay? And up here in the arms, there's little chains, like 2 little chains, okay? And it's up here at the tip of the arms, that's the variable region. This FC region, this is the stem region. It is conserved within a species, and we will see that that stem region, that triggers macrophage, that's going to trigger complement. Okay, and we had a nice conversation today, a nice presentation about impetigo, okay, and about how Staph aureus has they call it a protein A, but what it is, it's a receptor for FC region. So they bind antibody at the stem. You'll see that will mess us up. Okay, that's a trick on their part. So the FC is what complement is going to bind to, phagocytes bind to. This is an important portion of antibody. Okay. It's at the variable region where we have the little nooks and crannies. So what this is trying to depict is that this green antigen fits perfectly in this particular antibody, but it doesn't fit into, other ones don't fit into that little nook. Okay, so it's a very specific binding there based on shape. So there's a lot of variety, a lot of variable regions. We most likely have made antibodies for something that doesn't exist, okay? A nook and cranny for something that doesn't actually exist, okay? But if it exists and it looks somewhat like it and triggers it, we will get the responses that I'm about to say. So an antigen, an antibody generator, is typically a protein on the surface of a virus or a bacterium, or it could be yeast. And this protein, it in itself has a shape and has twists and turns, okay? And so the little bits are called epitopes. I don't need us to know this terminology, but I want you to see something, that if this is the antigen, let's just say that's a spike, that spike actually has this part that's up here to which I can make a blue antibody, and a part that's down here to which I can make a red antibody. And we're not showing, what about this stuff? Sure. And this line here, yeah. So I'm going to create a ton of antibody to one antigen. And when we're thinking about a virus, especially an RNA virus, and just, you know, how does it replicate? Well, it uses replicase, right? 0 proofreading. It's gonna mutate. It's not gonna look alike. Well, okay, did you change 100%? That's impossible. Then you can't land on the receptor, you can't get in. Okay, so you change this blue stuff, doesn't look the same, that's fine. These antibodies are no good, the red one still understands. Okay, so what I'm trying to say is sometimes it's depicted in a cartoon like one antigen, one antibody. I can make a ton of different antibodies to that same antigen, okay? All right, so that's more on that. All right, and the other thing is that there's parts of this antigen you see that are sort of buried within the fold. See how these lines here, okay, they might not be on the surface of that protein, but rather buried within. Well, guess what? Even that can get shown. Because this antigen brought inside of a macrophage or something, chop, 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 pieces get shown. Pieces on the surface, pieces in the middle, all over the place. In other words, I can wind up making an antibody to every last bit of this thing. Okay, does that make sense? All right. So we'll just remind ourselves about MHC class 1 and 2. That 1 is on every nucleated body cell, which means it's not on red blood cells. Okay, we've established that already. At all times, all cells must display what are you up to on their major histocompatibility complex. I mean, I don't want to spend more time on this really, but this is very, very nuanced to each person, and this is why you can't accept an organ from somebody else, because bam, the MHC is all wrong. Okay, so stop right there on that conversation, but all nucleated cells at all times must be displaying what are you doing right now. MHC type 2 though, those are only on antigen presenting cells, and there's only 3 kinds. Macrophage, dendritic cells, and B cells. That's it. Those are cells whose job it is to take, swallow, and display. That's it. Okay. The 2 types of T cells, T helper cells, and cytotoxic cells are different in their scope and what they respond to, and also what receptors on the surface. So they both have a T cell receptor, but one has CD4, one has CD8. Okay, we remember that this is the exact spot where HIV binds, right, to that one, so it doesn't bother T cytotoxic, but yes, it would bind to T helper cells. T helper cells are going to respond to MHC type 2, T cytotoxic to MHC type 1. Okay, big difference there. T cells and B cells have a lot of things in common. Where they were made originally, bone marrow, absolutely, but they're going to mature in the bone versus the thymus. What's on their surface? B cell receptor, T cell receptor. What about, can I find it in the blood? Students always mess this up and they tell me that you can find B cells in the blood. You can, really low numbers. If you see something that looks like a lymphocyte in a red blood cell smear, you're looking at a T cell most of the time. Okay, B cells really, they live in the nodes and they live in the spleen. Okay, they're not typically in the blood. T cells literally can be anywhere they want. Okay, receptor for antigen. Well, the B cell receptor, that's what it's for, and the T cell receptor will understand antigen if it's presented on an MHC. That's an absolute for the T helper cell and the cytotoxic cell. They don't understand antigen. It can fly right past them. They don't understand. It has to be shown to them on an MHC. That is not true for B cells. B cells will respond to little bits of antigen. You'll remember in the phagocytic event that part of what happens is, yeah, the thing gets caught, okay, it's got some smelly bits, the pathogen-associated molecular patterns, and then the pattern recognition receptors would find it. Okay, fine, gets brought in, endosome, lysosome, smash it all up. Okay, it's part of it on MHC type 2, but what happens to the rest of it? Egestate, right? All those little bits. B cells can respond to all those little bits. They don't need to have something shown to them. Okay? They can respond on their own. That's a big difference. So B cells will end up making antibody. T cells, it's going to end up killing abnormal cells. Unfortunately, also involved in hypersensitivity reactions. We talked about superantigen being a big problem when it comes to T cells. Okay. self and non-self, big difference, and important that we don't respond to non-self. During maturation, okay, if there's any in all of these T or B cells that are responding to self, they will get eliminated. That's known as clonal deletion. So they should get deleted if they're responding to self. And this will happen in the bone marrow or it will happen in the thymus. That's what's happening during their training. Once they go and live in the lymph or the spleen, they're just sitting there. And when an antigen binds, that one gets triggered to proliferate, to divide. That's called clonal selection. And so because a stimulated cell, when it divides, will do mitosis, and mitosis produces identical copies, that's a clone. Okay, so that's clonal selection. Okay. So this is our third level of the immune response. And so let's remember that if we were to introduce pathogen parenterally, okay, that would immediately, I mean, of course, so you stab yourself or step on a nail, it's got bacteria. No way around it. The mast cells will get triggered and I get inflammatory response, absolutely. But you also happen to have resident dendritic cells. Dendritic cells, they're just like a macrophage. They're kind of like cousins. They've got long processes, right? And they have pattern recognition receptors. They can bind the pathogen. So they'll bind it and chew it up, spit out some, and display. They will get caught up in the fluid, the interstitial fluid, and get caught into the lymph vessel. Remember that the lymph vessels have valves to prevent backflow, right? So this interstitial fluid goes in here where there's waiting T cells. They're called naive because they were just made and they're just now waiting to get triggered, just waiting. Once they get triggered, they get activated. Now we begin the third arm of the immune response. So these are the dendritic cells binding to pathogen, and then they will migrate in. You know, the pictures, I'm sorry, they're just not to scale, obviously, okay? They're smaller than the vessel. So they just get caught in there. Now, how is this fluid being moved in here? That's right, skeletal muscle contraction propels this fluid. And what you're really doing when you're moving your skeletal muscle contraction like this, you're letting yourself sample what just came in here. You're sampling it. So it comes in, and the inset shows us that here you're having this dendritic cell displaying on its MHC type 2 to the T helper cell. Look at what I just ate. Look at that. What do you think? Okay, that's what's happening right there. And we remember that's their role, display it on MHC2, but then look at all these little pieces. Okay, all those pieces can also stimulate. Dendritic cells are not the only ones that can present. Remember that macrophage can do it, and so can the B cell. So these little bits that just came out, remember that in the lymph node, that's B cells are there too. They have their B cell receptors, 100,000 all over the surface of any given B cell is just waiting there. If one of those little bits triggers that B cell, it will bring it in, it will display to the T helper cell, which is sitting right next to it. Hey, look at what just swung by. Okay, so it also can display. T helper cells, they respond to things on MHC type 2, but T cytotoxic will find an infected body cell because it will display what's on its MHC type 1. Okay, so I'm going to skip past that. Once a T helper cell is stimulated, okay, it bound something on an MHC type 2, all right, it's wrong, I don't like what I'm seeing. It speaks with cytokines, a lot of them. And those cytokines are going to have multiple effects, which I'm going to ask you to know a few. Okay, now I'm not sure how I'm going to, I want you to just familiarize yourself with it, so all of the following are true, or, you know, which of the following is true, something like this, okay? But so speaking with these cytokines, okay, there it goes. Some of them are going to be to stimulate T cytotoxic cells, go find anything that is displaying something wrong, or stimulate B cell, go ahead and get started in making antibody. I want you to recall at this point here though, that the T cell receptor and the CD4, okay, they're just like momentarily docking on the antigen presenting cell, and then they should let go. If you have a super antigen, that would hook these two together, okay, and not let go, so that we get the continuous spitting out of cytokine, which leads to inflammation, nausea, vomiting, fever, fluid loss. What's next? Shock, and then death. Absolutely, yes, you just jumped to the,
Yeah.
a little shock in there first. But, so, like, I mean, I'm trying to like put all these thoughts together so it's just like one thought about what's happening here. So if you could draw yourself, you know, maybe a dot, right here, like if there were a super antigen, that's what that's doing. Okay, like keeping this dock down here, and no, you can't let go, okay? Dendritic cells can themselves get sick. Is anybody doing mononucleosis? Okay, so you've heard of mono. So mono is a virus, Epstein-Barr, and it will preferentially infect B cells and dendritic cells. So a dendritic cell that is sick can actually say, I'm sick. It's not, its only job isn't, you know, to find and destroy pathogen, or not destroy, but display pathogen, but rather it can also say there's something wrong with me. So that can happen. Dendritic cells, because they have T helper cell, MHC type 1 and 2, they can trigger T helper cells or T cytotoxic cells, both of them. Okay. Once a T cell, which is, it's termed naive before it gets triggered. Once it gets triggered by that dendritic cell showing it, okay, I just saw something, it becomes an effector T cell. And this is what I'm saying would be, I think, a multiple choice here, okay? Lots of things can happen. First off, it can activate, and of course it speaks only with cytokines. So it can speak with these cytokines to activate macrophages to enhance killing, like work a little faster, okay? But it also works to kill chronically infected cells, okay, releasing bacteria. So any cells that have bacteria in them quietly, okay, can get released. Chlamydia, okay, that would work here. Get that stuff out, spit it out, let us have a fresh look at it. Okay, that should happen. It also puts out interleukin-2, and it's going to alter how many T helper cells versus other cells that we have, and so more cytotoxic T cells will be made. Okay, so kind of flip the balance, get me some killers out here. It also tells the marrow, I need more monocytes. You know that monocytes will differentiate in either to macrophage or dendritic cells. It allows more diapedesis to happen, so capillaries get leaky. And we said that just a second ago, that was one of the problems with super antigen. Okay. It also is like chemotactic. It gets macrophage to come kind of get over here and help clean up this mess. It also, and this is the last one I just cut and pasted over here, so this picture is over there, but this one here, it's going to have the B cells start dividing faster to become a plasma cell, which makes and secretes antibody. So that's a lot of stuff that it's doing. Okay, so just sort of familiarize yourself with this list, okay, of all the things that it does. Cytotoxic T cells, okay, what they're going to do is kill a particular infected body cell, anything displaying a foreign antigen on its surface. And it will do so by associating with the MHC type 1, and what this picture is supposed to show you is that, okay, here's the nucleus, and near it is the rough ER and the Golgi. All of that stuff, the rough ER and the Golgi, starts moving, the actin filaments move those structures over here right up against the infected cell so that we can make and secrete these proteins. Very important proteins: perforin to make a hole, and granzyme to go through the hole and trigger the apoptotic event. Apoptosis is a programmed cell death. I know that images of cytotoxic T cells will make you believe that the cell lyses open. It's not that. They get small and die, kill yourself. Do you remember who cleans up an apoptotic cell? A macrophage. Right? It's such an important cell. I talk about it continuously. Really important. And just to go back here for a second, that's one of the things that a stimulated T helper cell does, is tells the marrow, make me more of these guys. They're so potent. Please make more of that. Okay. So here we are at natural killer cells, which actually belong in the innate. But because it involves the MHC type 1, I left it to now. And they're very similar to B and T cells, but they are part of innate. Natural killer cells are cells that will just kill any nucleated cell, has to be nucleated, any nucleated cell that does not display MHC type 1. All cells at all times must show on their little flagpole, MHC type 1, what they're currently making. If you are not showing me an MHC type 1, that's suspicious and you're going to die. And they do it in the exact same way as a T cytotoxic cell. They secrete perforin and they secrete granzymes. This cell is going to undergo apoptosis. What kind of cell would stop displaying MHC type 1? Well, cancerous cells, you'll remember, they don't do G1 and G2 anymore. And you might remember me saying that a cell that has just divided is going to spend a fair amount of time in G1. That's where they're going to differentiate and start expressing particular proteins, including MHC type 1. When we don't bother with that anymore, because all we're doing is copy the DNA divide, S and M, right? That's what the cancerous cells do. Then they're not displaying MHC type 1, and that in itself is a trigger for natural killer cells. I would love to believe that this is such a wonderful system, that there's no such thing as cancer, and yet we know that it's the number 2 cause of death. So though this is the original reason behind it, and maybe it would work great in systems from the past, okay, it's not keeping up with our modern life. I mean, there's many things to be said about this. How long did people live? You're much more likely to die from some other random accident type thing. I'm sure accidents were top 10 back in the day. But, they're still in our top 5. Okay, so let's not kid ourselves. But I think it was higher up on the top 5 than it is currently. We can't explain half the reasons for our cancers. Not even half, what am I saying? 90% of the reason for our cancers, we cannot explain it. Okay, so that's the original purpose for this. Obviously, it doesn't work well enough. Okay, all right. So now this is, I have a slide where I want to. That's coming up. I'm gonna stick to what I wrote on the thing. Okay. B cells, which are in the lymph or spleen, and you'll notice there's different B cells here, and they each have a different colored antibody. This is just to show us some of the possibilities of the 10 to 14 to 10 to 18 different antibody types that could be on the surface. Okay? Each with a different variable for each. The stem would be identical. Okay? The antigen that binds to this one stimulates this one to start dividing. It will divide once, twice, and this is a cartoon image, so they're skipping to the end. It takes many, many more divisions to get to this final layer. In fact, this layer will stop. These guys continue to divide a little bit longer before becoming plasma cells. Let me ask you, how long does it take this cell to divide?
20 hours?
24 to 26 hours. Why so slow? What's the deal?
The specificity of the antigen.
No, it's not this bad. I mean, why? I mean, even without an antigen, why can't any given cell of mine divide faster than 24 to 26 hours? What's the deal?
Duplicate everything.
Yeah, lots of DNA, lots of organelles. It's not that simple. Okay? When you're looking at, okay, I'm trying to duplicate one circular piece of DNA, that's nothing. Okay, this is, you have what, 46, right? That's a lot. So we spend a lot of time gearing up. We have a cell cycle, all of this stuff. Okay, so that's 1 day, 2 days, many more days than this to finally get down here where we have memory cells, and honestly, these would continue to divide further for longer before they become plasma cells. If you are looking at these cells, you can see they don't look the same as the initials. I see the nucleus, that's fine. What do you think this is? That's right, it's ER and Golgi. Why? Because this job of this cell is secrete antibody. The story of the secreted protein is one that we have told many, many times. I left my conversation and discussion about eukaryotic cells very, really, really simple, but I spent a lot of time right there because I said this is what immunology is about for us. The secreted protein, whether it's interferon, whether it's complement, okay? Those are secreted proteins. They're made in the exact same way every single time. Through the ER, sugar slapped on it. Then to the Golgi, more sugar slapped on it. Eject. Right? That's this. These plasma cells, once they get going, and I have to look this up every semester because I just will not stick in my head, 10,000 antibodies per minute. Okay, so that we have to just pause for a second and ask ourselves, is the making and secretion of an antibody, is that anabolic or catabolic? Oh, that's anabolic. So use ATP or yield ATP? Yeah,
Oh,
use
it
it, yes, like major. So, I mean, if you want maximal immunity, you gotta yield the ATP to it. I can't also be asking a lot of myself or be under a ton of stress or be trying to solve some other issue like a broken femur, okay? Then I'm immunocompromised. We have to just recognize this is a potent system that is going to eat energy, okay, if you want it to work properly. Okay, so the B cell can be stimulated all on its own, but it can be helped if the if it shows the T helper cell, look what I just ate, the T helper cell will secrete cytokines and that division will go a little bit faster, okay? So T cell and B cells do interact because a B cell is in fact an antigen presenting cell. It's a short list, there's only 3, macrophage, dendritic cells, B cells. So they can bind with their B cell receptor, which happens to be an IgD, bring in the pathogen, break it up and display. Okay, they can do that. Okay, so that's what's happening here, all right? They can be activated independently, that's fine too. All right, so this is my picture. No, not quite yet, okay. So here's the activated B cell, only this one gets activated. And many, many, many divisions. I like this picture better because you see how the memory cells kind of just are a subset that stop dividing while the rest of them continue on. Okay, for more divisions, that's a better picture than the one before. And then those plasma cells, they're going to secrete antibody. This picture shows it suddenly in the blood. How did that happen? So let's think, where is this B cell? In the lymph node. And so where's all this division happening?
Lymph
In the
nodes.
lymph node. What's happening to my lymph node?
Swelling.
It's swelling. Okay.
Infection.
It's swelling.
Get swollen.
Right, you get swollen lymph, and all that tells me is whereabouts this whole thing started, because lymph nodes drain from certain organs. Okay. Then this plasma cell, which is in the lymph node, is now secreting antibody. That's in the lymph. How does that get into the blood?
Through the trunk.
Thoracic duct. How quickly will it get there?
Not very.
Well, I mean, it depends on what? Of what?
Where
Skeletal muscle movement. I can't take the body because it's too vague. It's too vague. We had to like pin it. Okay, so skeletal muscle contraction. Okay, like rapid eye movement, is that gonna count?
No.
No, okay, skeletal muscle contraction. Okay, so I can't will it there. Okay, I don't think. So, that's how that happens. What does the antibody actually do? Very important to understand, the antibody does not kill the pathogen. An antibody is a little tiny protein. It's like smaller than the pathogen by a lot. So it can't kill them. But what it can do is, because it has two arms, drag them out of solution. Okay, that's called agglutination. If they grab them and they start falling out of solution, they can't stick to the cells that they would like to stick to. Okay, remember that's part of pathology. Come in the right door, in the right number, stick. Can't stick? No good. Okay, them binding to pathogen, notice it's the variable regions that binds. We already said it would be specific to a particular pathogen. Notice that the stem is sticking out. That stem is attractive to complement. This is actually called the classical form of complement activation. I'll leave it to the physio to hammer that into you. There's 3 different activations. It doesn't matter to the microbe. I don't care how you triggered. You got now complemented, because now 3 things happen to you with complement. You can't pick and choose. Opsonization. That means that the macrophage is going to come over here and find it, because macrophage has receptors for not only complement, but also FC. You see this here? Okay. But we get opsonization, inflammation, and the MAC attack complex. And of course, that's the only one they want to show you. Okay, MAC attack complex. FC region by itself is enough to trigger phagocytes. They have receptor for FC. But you'll recall, I just said it 3 minutes ago, Staph aureus has a receptor for FC. So here's the antibody you made to Staph, and you want to try to grab it, and Staph's like flipping it around so that the variable regions are sticking out, not the FC. Macrophage doesn't understand variable regions. How's it going to understand 10 to the 14th to 10 to the 18th different things? It can't see that. It's trained, right? Look at this, FC, that's it. This is a flip this whole thing around, okay? Guess what? That's a short list. It's not like every bacterium in the world knows to bind FC. No. Certain ones, right? Pathogens. If you're a pathogen, something special going on with you, okay? Because other bacteria, you get caught. You're smelly. Here's my antibody binding. Here comes complement. Here comes macrophage, okay? Here's one. There's a particular kind of antibody called IgE. It binds to parasitic worms. And the FC region of that is bound by eosinophils. Eosinophils are part of the granulocyte class. They have little vesicles in there that are going to release, but what they release are lytic enzymes, meaning that they lyse plasma membranes. A worm is too large for a macrophage to swallow. Okay, a worm is a multicellular organism, not too big. Okay, they're microscopic, but it's too big for a macrophage. So instead, the way we handle it is to squirt out all these lytic enzymes, pop holes in the membrane. But a worm is an animal. Animals don't have walls. That's just like me. That's my thing, right? So Remember I said that eosinophils are implicated in a lot of allergic responses? Okay? There's not a lot of worms out there. They're kind of bored. A triggered eosinophil is dumping lytic enzymes. You're destroying your own tissues. Okay? Eczemas, that's what's going on there. You're destroying your own tissue. Why? Not sure. We don't have an answer for this. Right? But we do know that's what's happening. I just don't know how to make it stop. Okay? Other than, okay, blanket, steroid, everybody, stop. That's not great. Okay. All right, but then this one, neutralization. Arguably the most important thing that an antibody does for us. Antibodies will bind to the surface of virus and bacteria, and because virus in particular need to land on a receptor with their spike to get in, if they can't, it's over. A virus outside of the cell, it cannot propagate. It cannot make more copies of itself. That's not possible. It doesn't have ribosomes, it doesn't have ATP generating, right? Like, we already established that. Not happening. What about the bacterium? Well, it doesn't necessarily need to be inside the cell. There's certain ones that do. Chlamydia likes to live inside the cell. Not all of them, okay? All right, what about the rest of them? They have to stick, because if they don't, I got peristalsis moving you out. I have my turbinate moving all the time, right? I have all of these ways to, like, I'm sloughing. What's your plan? You've got to be able to stick. Without sticking, it's not happening. Their pathology cannot occur. So that's huge. But even bigger is this, the binding of toxin. Endotoxins, not so much, because those are lipids and have no shape. I'm speaking of exotoxins. Exotoxins are secreted proteins. Very few bacteria, of all the known bacteria, can make an exotoxin, but those that do, it's quite serious. Botulinum, 1 to 3 nanogram per kilogram. This thing is toxic. It has to bind to its tissue type. In this case, with botulism, it would be the neuron, right? Okay, but other exotoxins, like ciliary exotoxin from the whooping cough, it's binding to the ciliated cells. Can't do it if antibody is all over the surface of it. You can take an antibiotic and kill the bacteria that makes the exotoxin, but what about the exotoxin itself? How are you going to mop that up? You need to have an antibody. If you don't have that, and it is something very serious, like botulinum or tetanus, you got to get somebody else's. Okay, I cannot wait as long as it's going to take for you to ramp up and divide and divide and divide, right? It's going to take too long. You need to have somebody else's antibody. They'll call it the antidote, okay? But it's somebody else's antibody. All right, so very important. These are the 5 classes of antibodies. And, you know, it's just me and my regimented thought. I have to start with A, I'm sorry. So, all right, IgA. IgA doesn't even really belong in the specific immunity because it's part of innate. It's just secreted with all kinds of mucous membranes, and it's in essentially almost all of our bodily fluids. So it's in saliva, it's in your tears, it's in your intestines, in the blood, it's in mother's milk. It's just a random antibody out there just in case. Why just in case? Because bacteria are everywhere. Your likelihood of encountering it is, 100%. Like that's the way you're made. Remember I said this, that your entire immune system is designed with the knowledge they're out there. That's all there is to it. But you're ready. I have all these bodily fluids and I'm just like randomly putting out IgA. There are bacteria that have IgA proteases, they'll destroy it. Neisseria, gonorrhea, and meningitis has this ability. Let's get rid of it. Okay, those are organisms that want to live in particular types of mucous membranes, and they have a way to do that because they can kill or destroy, I should say, your IgA. Did you have a question before I go on?
I was gonna say, what exactly is, like, I know it says it's the structure, the dimer structure?
So it's 2, see how there's 2 Ys?
Mm-hmm.
That's what it means, dimer, just two.
And it says it has a
Secretory component, these are sugars.
Okay.
Yep, these are sugars.
How does that help? The sugars?
Helps?
Yeah. how
Okay,
does
so
that help?
sugars help in many ways. They are protective of the protein itself. They're also help with solubility. Okay, so if you think of having any kind of polysaccharide like breads or crackers and throw it into water, it takes up a ton of it, doesn't it? And suddenly a lot heavier than it was, right? So this is like, it makes it a lot more soluble in aqueous environment. Okay, so that's A. D would be next. IgD is actually the B cell receptor. And if you think about that, then the FC region is actually buried in the plasma membrane, okay? And so that's very little of the antibody out there. In fact, if it's ever found, it means that there's a dead B cell around because it shouldn't be loose. It should be part of the B cell, okay? IgE, okay, this is the one that gets bound to mast and basophils cells throughout the body, allergic reactions, the lytic of parasitic worms. It's kind of a bizarre one, but there you go. None of these fix complement. Complement wants to see the FC. The FC here is not visible. They face each other. They have a secretory component over the surface of them in this little linker region. IgD is buried in the membrane of the B cell. Okay? IgE is really about eosinophils, so this is not a complement thing. Neither do these things go through the placenta. None of these. Okay? So A, D, or E. When we get to IgG, this is the antibody. This is the primary antibody you're going to find. 80% of the serum antibody, it's IgG. Antibodies that we have are proof that you have encountered a pathogen, either for real, on your own, or through vaccination. Okay, so we could, back when they were saying everybody had to get a COVID shot, right? Okay, you had to prove it, to show whatever. You could prove that some people did, they didn't want to get the vaccine, already had COVID. They just take their blood, check for anti-COVID antibody. Yep, you have them, you're good. All right, that's proof. We will find IgG in blood, lymph, and intestine. It has the longest half-life, so it lasts 23 days. In 23 days time, you have half as much, and 23 days later, half again. So it doesn't go to 0 in 23 days, it sort of tails off. It does fix complement. In fact, everything I just said a moment ago about what antibodies do, including fixing complement, it's a discussion of IgG, and it's the only one that goes through the placenta. All of mother's antibodies that she has ever produced, when she got sick with the mumps at age 7, and the cold she caught last week, all of this is being passed through the placenta to the baby, all of it. Okay? It's also in mother's milk. All right. But the very first antibody made is actually an IgM. When we first trigger, okay, an antigen lands on a B cell and it goes through its division cycles to become memory cells and plasma cells, the first thing it makes is an IgM. And this thing is huge. It's 5 antibodies, basically. And it is so big that there's no way that this can get through the placenta. It's just too big. Okay? It doesn't really last that long, but it actually is the best one at fixing complement. I recognize that here they've flattened it out, but you have to think of it as like 5 legs sticking up. So it'd be like at a peak. Okay? So it's like hyper attractive to complement. They come right over here. Okay, IgM. So, and it's the best one, of course, at agglutination because it doesn't have just 2 arms, it's got 10. So it's dragging a ton of stuff out of solution. Whatever it is cannot bind, for sure. Okay, so here's my picture to draw the lymph and circulatory connection. So, I drew part of this the other day, but we'll just do it again. So we're clear. Okay, so this is your tissue. capillary system, and we said that it's under pressure on this side, the arterial end, and the venal end under less pressure, and that we have some fluid loss here, some goes back, that this is called interstitial fluid. And what I maybe failed to say last time is that the lymphatic system has two functions. And the two functions of the lymphatic system one, immunity, sure. But two is to drain interstitial fluid. And bring it back to the vein. Okay, so during some surgeries, they will remove lymph vessels and lymph nodes, and then afterwards, depending on what part of the body it's in, sometimes people will get edema, which is just the buildup of fluid, because if they, remove that lymph node because maybe it was cancerous or whatever, now that area could potentially get edema. And this is people wear, compression sleeves and this kind of thing. That's what that is. Okay, so when we have a parenteral entry of pathogen, you also have here your resident dendritic cell. And it's true, you would also have your mast cell. Okay, which is part of just the innate response, and the mast cell would release the histamine, and the histamine would make this leaky, and the neutrophils would come, and all of that stuff that we already described, all of that would happen absolutely right away, and in the same exact format, regardless of which pathogen came in this time. Okay, it would be the exact same thing that happened last time. But now, if this dendritic cell has swallowed this pathogen, we know that it will display on its MHC type 2 a little bit of that pathogen. Now, you just have to, suspend your disbelief here, because if the pathogen's that size, it's not going to be the entirety of it. This is the MHC displaying MHC2, and then this is displaying a little bit of cancer. And it gets dragged in here. My picture's not much better because this cell would be like that big. Okay. And it gets dragged in here. Okay, skeletal muscle contraction would propel that fluid in there. Okay, in here I have my B cells. And each B cell has a unique IgD. I have 10 to the 14th to 10 to the 18th different kinds of shapes that I can have, but I'm making the shapes the way I know how to make a shape. All right, so here it comes, and remember that not only is some of this going to be displayed, but some of this is going to be egested. Okay, so that little egested bits are also flying down here. And I'm going to keep my discussion just to that egested bit, okay? So let's just suppose that it's this one that gets triggered. Okay, so that's the antigen. This is the IgD, also known as the B cell receptor. Okay. That one gets triggered. So now that one, we will start here, divides into two. Okay, that's 24 to 26 hours. And we have to do it again, and that's another 24 to 26 hours. And it happens again. It happens many more times than this. I cannot draw it out. I will tell you exactly how much time in a moment. I'm already losing count of how many circles I've drawn. Let's say, I'm drawing it too low now. Eventually, these divisions will give rise to this kind of cell, the plasma cell. Others would have stopped dividing back here. These are the memory cells. Memory cells will have that same kind of IgD on the surface, okay, because it's a clone. It undergoes mitosis. It creates the exact same cell, right? They have the same information. Okay, so those are the memory cells. These become the plasma cells, which secrete initially an IgM, but after a couple days, it'll class switch to IgG, which is the main antibody, so I'll just do that. And all of this is happening in here, so this is getting much bigger. Okay? And when finally these plasma cells start making and secreting the antibody, they're here in the lymph, which joins up at the thoracic duct with the vein, and now it's in the blood. And now it's throughout your body within 1 to 2 minutes, right? Okay, so this is a big deal. This is how this works. The problem is that we have this constraint, that we are huge. There's a lot of information to copy, a lot of organelles to subfractionate, and it takes a while. This is not my only immunity. I have my whole innate response happening, right? This would have happened right away. Cordon that thing off, try to eat it up, okay? And all in the meantime, this is happening at the same time. It's just that this story is taking longer to get started. Yes?
Are we gonna draw the thoracic duct? The connection between the circulatory and the lymph system?
Well, I'm not very good at this, to tell you the truth, so let's, so this is somebody's face. Okay, I don't know. It's right in this zone here. Okay, I'm sorry, I'm not good at this. I'm just not. There's a better picture. There's a better picture, maybe, it's maybe the third or fourth image on this PowerPoint. Okay, where it sort of shows you where they go together.
Mm-hmm.
Okay.
Are we gonna have to draw that? Or just this?
I try to minimize drawing on an exam. Some people will, why? Because some people, this is going to be 3D. And it's you just give me the information. I don't need great detail. But I might have that. Right? That's an important understanding right there. But so here we are. And so once we have these two populations of cells, these are the ones making and secreting antibody 10,000 per minute, but we have this set of memory cells. And these memory cells are what explains the secondary antibody response. This is the terminology. How can you explain the secondary antibody response? That's what I'm about to describe. So the secondary antibody response is because of memory. So remember that these cells are going to be here. They have the same exact IgD that got triggered in the first place. So the first time you get challenged with a pathogen, it takes almost 2 weeks to get maximum antibody response. 2 weeks to an organism that can divide every 20 minutes, that's forever. Okay? Thankfully, that's not all I have going for me. I've got my fever response, I've got my inflammation response, I've got my antimicrobial peptides, I've got my complement, I've got my transfer and hiding, right? I'm doing other things while this gets ramped up. Okay, so about 2 weeks for the response, and over time, after 23 days, I got half as much as I had, right? Okay, it kind of goes away, never to zero, but just half and half and half again. Okay, the second time you see this thing, in about a day and a half, do you see how much more you get here? You just need 1 or 2 more divisions to get yourself to the plasma cell. Why? Because the memory cells are almost all the way there. They did most of the division already. They're just waiting for another trigger to, okay, here we go. And now they're that. You're not starting from scratch. That's the idea. So that you have a tremendous response next time. So, what does this mean? Remember back a long time ago, who's going to get sick, who's not going to get sick, and then I had this image that maybe was a little bit confusing to look at, that guy on, like, I don't know, the subway or the bus, right? Like, who's going to get sick? And it had to do with, well, what kind of pathogen is it? Did it come in the right door? Did it come in the right number? What was your overall health like? And one of the questions was, did you see it before? Because if you've seen it before, then I got this coming at you. So it's not that you've not been exposed to something you've seen before, it's that you mounted your secondary response, which dwarfed the first one, okay? However, if you're seeing something brand spanking new, you're starting from scratch. Your question, Palm.
If their half-lives are not that long, how are they staying then in our system at that low level for a long period of time?
They are there for a very long time. It takes, to get to zero on a half-life, it's a while. It's not right away.
Because there's so many.
There's so many.
Okay.
So it's if it's shooting out, let's say for 3 or 4 days at 10,000 a minute, right? I mean, it's a lot. That's a lot. There's a bad thing to this, though. Let me answer your question and then go ahead.
So mine was kind of going on like the COVID-19 vaccine. So I got vaccinated before I got COVID, and then I ended up getting COVID, and I was out for like a good 10 to 14 days, like really bedridden sick. Is getting like, so the whole point of getting like a vaccination, stuff like that, is to like basically lessen the chance of getting sick and also lessen the severity of the sickness. Does that mean that theoretically if I weren't to get that vaccine, I would have had a much harder time with getting sick?
That's what they say, but the thing is, half the time with vaccinations, you're kind of guessing, especially if it's an RNA virus. Because the thing you're creating right now, this is, so the flu, Judith. Okay, so I asked you what was the efficacy of that this year, because the problem is almost every flu begins in China. So they go to China, figure out the flu that they're currently dealing with, they start growing it in eggs, and you create antibodies against this flu. The problem is, while it's making its way across the globe, it has 8 strands of RNA and replicates making the copy. It's making mistakes, one after another after another. So by the time you're getting the vaccination, it's, how effective is it? This isn't the same guy anymore. And so initially, in particular with the COVID, it was kind of hit or miss. Okay, we're a lot more comfortable with bacterial vaccinations and DNA virus vaccinations because the likelihood of mutation is less. Okay.
One more question about
All right.
vaccinations. Sorry. So I also know the Johnson & Johnson vaccine, when they first came out, they were having a lot of issues due to it causing blood clots. Why would that be if it's a vaccination?
Because the Johnson & Johnson, they took an adenovirus, okay, a naked DNA virus. They pulled the guts out of it. So the DNA belonged to the adenovirus. They took it out and put in the DNA for the spike protein of SARS-CoV-2. SARS-CoV-2 is an RNA virus, but DNA lasts longer. It has more, shelf stable. RNAs, had to be cold, right? I mean, there's pictures of cars getting stuck on the freeway and then they're running around and who wants to get shot because it's like it's going to decay, right? So they did DNA. The adenovirus is not a harmless virus, okay? It has its function. It grabs onto particular cells. For some people, it was causing agglutination. That's it. So we're guessing half the time. All right, so this is what I want to say about the secondary response, though. Okay, so it takes 2 weeks for this one. This one within 1 to 2 days dwarfs the first one. They shouldn't even be on the same scale. Okay, that's great. That's fantastic because you see the thing a second time. But let's pretend that it's not a pathogen. It's shellfish. And your body's absolutely convinced that this thing is wrong, and you made antibodies to it, and you have memory cells too, and the next time, wow. Okay, so little you know, like a little too strong and against the wrong thing. So we do have to watch ourselves. If you have a shellfish allergy, a peanut allergy, really don't go there, because you're just waiting. Those memory cells are just they're just waiting, okay? And they will get triggered. So that's a problem. Normally though, for most things, I'm so happy for those memory cells. You know, this is why you can be around people who've been sick if you've had that, because you're already all set. Okay, so initially, our initial exposure, you see the IgM is first and the IgG quickly takes over as the main antibody. And the same thing will happen next time. You'll make a little IgM thing, but really it's an IgG response. Okay, so we have memory, and that's a big feature of the adaptive immunity. We said that it was slow, specific, and had memory. Okay, so why is it slow? It takes a really long time for these cells to divide to get to the point where they can actually make antibody. Why is it specific?
Because it has the receptors of the antigen.
Well, the antibodies that are made were exactly from a receptor that bound to that antigen, so it has the affinity for that antigen and only that antigen. So it's specific, and the memory is because of the memory cells. Okay, so we like, rather than just, okay, this is the list of characteristics, like, can you explain them? Right? Okay. All right. So they're gonna last for a long time, and the thing is that there's more IgDs, 100 times more, which is kind of ridiculous because it's already 100,000. So more than that now on the surface of the memory cell. In fact, it has a very limited function. It's just going to turn on if you see it again, but otherwise you're just kind of sitting there. You don't do much at all. They have more MHC2 on their surface, so they can get the T helper cell help more faster, and so they generate plasma cells faster. It's an awesome thing to have this memory. And so this is terminology that I'm really sorry, but you do have to know it. I've seen that it's asked on NCLEX and junk like this, so I'm sorry about the word cited. I'm about to say. So here's your acquired immunity. You can get it naturally or artificially. Artificially means clinically, okay? In the real world, that's natural. So you can get it in an active way. And that would be to actually encounter the pathogen on your own. You breathed it in, you swallowed it, it got, you know, parenterally introduced, whatever it is, you saw it, you responded, your B cells triggered, divided into plasma and memory cells. Okay, you made antibody, you dealt with it on your own. That's active. A passive form, which happens in real life, is the baby getting antibodies from mom, either through the placenta or through the milk, mother's milk. The problem with passive immunity is that it does not give memory cells. It only gives antibody. Antibody is awesome. Okay, it has all those functions, neutralizes opsonization, triggers complement, all that stuff. But eventually, the baby starts eating real food and stops drinking mother's milk, and what happens to those antibodies? They decay in 23 days, and then 23 days, it's not just straight to zero, but it's, you know, it's on its way there. And so, if it's something that, like, you know, the mom gave it her antibodies to the flu, she had the flu prior or during pregnancy, that's fine. The baby is now exposed to it, no memory to that, none at all. And if the antibodies aren't there in high enough number, then the baby gets sick. Okay, because what it did not get is memory. So the difference between an active form and a passive form is active has memory. Passive only has antibody. It's absolutely protective. Very, very important. The thing is, though, let's say that mom is, you know, gave all the antibodies that she has to the baby, you know, and is currently doing that. They're nursing, okay, fine. But now here's SARS-CoV-2 that nobody saw. Mom's never seen it before. Mom's got to go through the whole motion. She's got to wait 2 weeks to get antibody response. During this time, in that 2 weeks time, the baby can get sick and mom cannot help it. Okay, that's the thing. There's no, memory here. And if the baby is under 6 weeks old, they can't even do it. They can't make their own antibody. Okay, so just, be careful taking your baby. Artificially, so this is clinically. This is the idea with vaccination, is that you will be exposed to the pathogen at a super low dose, well below the ID50 value. We know how much it takes to get sick from different pathogens, and you should be orders of magnitude below it. So if it takes 10 endospores to get you sick, and I was trying to give you a vaccination against this, it would have to be like tiny, tiny, tiny amounts. Right? You are not going to get a vaccine at the ID50 value, because then you're going to get sick. I want less. I just want you to see it. Sample it. That's it. Okay? And you'll go through the motions and, bind it and trigger the B cell, and it'll divide, and you get your memory and the plasma cells and the antibody. Okay, fine. And you get the memory. Artificially acquired is passively done when somebody is given another person or another animal, another vertebrate's antibody. This is done for things like botulinum. Right? If you've not seen this before, you're not vaccinated against it, and you're showing signs of it, I got to give you somebody else's stuff. They call it antitoxin, antidote. It's antibody. You're getting somebody else's antibody. I cannot wait 2 weeks for you to get started, not with something that has a fatality rate of 2 to 10 days. Can't go there. Snake venom. That's something else. You can get antibody to that. It's usually not a person, it's usually a horse. Okay? But I will say this, antibody is a protein. And if it comes to you from a horse, okay, you got bitten by a snake, you need to have this horse antibody, okay, that's fine. What does my body think about that FC region? Oh, that's not mine. And so I'm currently, okay, thank you for what you're doing, and I'm currently mounting my response against the FC region of that horse. So if this ever happens again, and I get bitten by something else, because it can't be that same one, because I got memory cells to this, but I get bitten by something else, I'm gonna go into the hospital, I'm gonna give you the horse antibody. No, not horse, can't have horse. Give me somebody else. Okay, go, fine. That's no problem. Some other vertebrate, no problem. Not horse, I can never see horse again. Okay, because you're gonna mount the major response against it, okay? So we just have to kind of keep track of what's been going on, what's happened, right? That, that's a rarity. I mean, for anybody to have to get somebody else's antibody, it's not that rare, but it's kind of rare. When back the first time we had President Trump, he got SARS-CoV-2, and they gave him everything there was to give, including somebody else's antibody. They gave him somebody else's antibody. Yes, they did, and it worked. And at the time I was teaching, and it was over the summer. It was a summer class, and somebody's like, "What do you think about this?" And I was like, "If we cannot save him, no one can be saved." And so when he was saved, I was like, "Okay, like we have the underpinnings of how to deal with this." I mean, it was early on. Now we know. Then there's different things we can do, but getting somebody else's antibody is a perfectly legit thing to do. Okay, but you got to recognize you are currently mounting a response against it, and just not for today. Okay?
Is it more that the antibody is itself is attaching to the pathogen, and you are attacking the antibody, the combination of the two? Is that the way that it's working?
Well, both. You are going to be making antibody against the pathogen on your own. It's just taking you too long.
Right.
And this antibody that's coming in, you'll look at that FC region.
Right.
And make antibody against that.
Okay.
So that you can't ever see that again.
So you're mounting a response to the antibody that you were given. Okay.
See, the thing is, we're capable of mounting multiple responses at the same time. And so it's not like, I'm sorry, I'm busy with this. You mount as many as you need to. So, last slide. Okay. So everyone's like, the last slide is this. It's just that sometimes we can have autoimmune disorders, and it's when, unfortunately, we respond to self. And so this response to self, unfortunately, can be we're making antibody against self, and we're also having a T cell that responds against self, leading to cell death. This is what happens in multiple sclerosis. You're killing your Schwann cells that are around your axon. This is what's happening in lupus. You're actually making antibodies against your DNA. That one's a really bizarre one. Rheumatoid arthritis. You think the collagen around your joints is hurting you. Why? Why are you thinking that? Like, what is going on? We don't know. Something happened. So no answers here. Only that we have such a powerful immune response that sometimes for people like this, your life is to be on a low dose steroid to just dampen your immune response. That's the only thing we know to do. Okay, so that's it. And